Our planet's oceans are just teeming with this hidden world, right? A kind of invisible chemical soup that holds a massive key to understanding the global carbon cycle. But there's a huge problem. When scientists all over the world try to look at it, well, they're not always seeing the same thing. Today, we're going to dive into that problem and how one really clever global experiment found a solution.
So what we're talking about here is something called dissolved organic matter, or DOM for short. It's this incredibly complex soup of thousands of different carbon-based molecules that you'll find in all water, and it plays a gigantic role in how carbon moves around our planet. Seriously, you can think of it as the chemical circulatory system for the entire Earth. Okay, so scientists everywhere are trying to decode this chemical soup, but that brings up a really big question. What if you sent the exact same vial of ocean water to 24 of the world's top labs? Would their super high-tech machines all tell you the same story? As it turns out, the answer to that question revealed a really fundamental challenge right at the heart of modern science.
Here's the core challenge, right? Each lab uses slightly different equipment, different settings. And this creates a kind of scientific Tower of Babel, where everyone is speaking a slightly different analytical language. So the question is, are we seeing real changes in the environment, or are we just seeing the differences between the machines? To figure that out, a massive global experiment was designed. So how in the world do you actually test if the entire global scientific community is on the same page? This is where the study gets really, really brilliant. First things first, researchers out in California created a set of perfectly standardized samples. They took DOM straight from the Pacific Ocean and they mixed it in very precise amounts with extracts from algae. This created a whole gradient of different mixtures. These would be their identical mystery samples. Then, they shipped these identical little vials to 24 different labs all over the globe. Every single lab got the same instructions. Analyze these samples with your own high-resolution mass spectrometer, but use the standardized method. It was a totally unprecedented interlaboratory test. Okay, so what happened when all that data came pouring back in? Well, at first glance, it looked like total chaos. The results were not what anyone expected, and they really highlighted that fundamental challenge we were just talking about. Get this, when they analyzed all the results together, the single biggest difference between the data sets wasn't what was actually in the sample. It was simply which lab the data came from. The lab-specific equipment, not the sample's chemistry, was driving all the patterns they were seeing. This slide shows you the problem so clearly. See, each dot here is a sample. The different colors represent the different labs, and the different shapes represent the different sample mixtures. And as you can see, the colors are all clumped together, but the shapes are scattered all over the place. The data was clustering by lab, not by the chemistry of the sample. It was a mess. The researchers pretty quickly realized the chaos was being caused by what they called noise. Thousands of these faint molecular signals that were being picked up inconsistently by the different machines. It's kind of like trying to listen to your favorite song through a ton of radio static, but they had a hunch that hidden within that static was a clear, consistent signal. And this just brilliantly illustrates the breakthrough. When they filtered out all that noisy static and focused only on the 100 strongest, most intense molecular signals, everything suddenly snapped into perfect alignment. Now look, the shapes, the sample types are all clustered together. The real pattern was there the whole time. So once the data was all cleaned up, a really clear biological story started to emerge. Now that they could finally see the true signal, they could ask, what were these reliable molecules actually telling them? This chart is pretty remarkable. It shows that for certain key molecules, every single lab saw the exact same trend. As the sample mixture changed, the signal for that molecule went up or down in perfect lockstep. It didn't matter which lab was doing the analysis, they had found their reliable markers. And these weren't just random chemicals either. They were known compounds that told a clear ecological story. So molecules like loliolide, which come from algae, they decreased as the marine DOM went up. And at the same time, compounds like cabryllostatin, which are known to come from Pacific seagrass, increased. This was the proof that the method could find real, meaningful biological patterns hidden deep in the data. This is why this study is so important. It does way more than just analyze some ocean water. It provides a clear roadmap, a blueprint, really, for how scientists everywhere can work together on these massive global challenges. The paper lays out 3 key recommendations. First, you've got to standardize methods. Use similar instruments and harmonized settings. Second,
Use quality controls, like common reference materials, just to make sure everyone's data is comparable. And third, share all the data openly, so the entire community can build upon these findings.
And this isn't just about studying the ocean. This is a model for any field, from climate change research to medicine, where the biggest discoveries are going to depend on our ability to combine huge data sets from labs all around the world. It's all about making sure we're all speaking the same scientific language. Ultimately, this study isn't just about fixing a problem. It's about unlocking potential. It really makes you wonder, right? What other profound discoveries about our planet are just waiting out there to be made once the entire scientific community is truly working from the same page?